You'd think the genetic code is written in the backbone. It isn't. The backbone is just a rail, identical the whole way down. The message is the order of the bases, and missing that costs more points than anything else here. 3 minutes. Let's build one from scratch and never mix this up again. It starts with one monomer, the nucleotide. 3 parts: a phosphate, a 5-carbon sugar, and a nitrogenous base. The sugar and phosphate are identical in every nucleotide. Only the base changes. DNA has 4 bases: A, T, G, and C. So the base is the letter. Sugar and phosphate are just the frame. Link them into a chain, phosphate to sugar, releasing a water at every join, like any polymer. That's the sugar-phosphate backbone, with the bases hanging off it. And it's monotonous. Sugar, phosphate, the whole way down. Same backbone, different base order, completely different message. The rail carries nothing. The bases carry everything. Now bring in a second strand. The bases pair across the middle. The pairing is fixed: A with T, G with C. It's not random and not like with like. A never pairs with G, so one strand specifies the other. Know one side, you know both. That's the secret to copying DNA. Each strand is a template. Here's what trips up the most people. 2 different bonds hold this together. Along each backbone, strong covalent bonds. They don't come apart. Across the middle, weak hydrogen bonds. 2 for A to T, 3 for G to C. That's the point. Weak bonds let it unzip without breaking the backbone. Glue the bases together instead, and DNA could never be read. Backbone strong, base pairs weak. The strands also run opposite ways: 5' to 3', partner reversed. A strand is directional. New pieces only get added at the 3' end. And running anti-parallel is what lets the bases line up. RNA is the close cousin. Single strand, slightly different sugar. And it swaps one base. Uracil takes thymeine's place and pairs with A. So RNA reads A, U, G, C. G and C don't change. Put it together and the structure does its job. Strong backbone keeps it safe. Weak rungs let it open. Each side rebuilds its partner. Stable enough to store the code, loose enough to copy it. So, 3 parts to a nucleotide. Bases are the message: A with T, G with C. Backbone covalent, rungs hydrogen, and RNA swaps in uracil. Get those straight, and Unit 1 nucleic acids fall apart. Want to step through this one yourself? Links below.